Within each cell, a sea of macromolecular complexes move and interact in a complicated dance. Understanding and explaining the subtle behaviors requires the ability to think in 6 degrees of freedom. Presenting them in publications and videos requires orchestrating the motion of thousands of individual elements, while avoiding collisions and showing their intimate interactions. The process of constructing a 3D space full of molecules currently requires learning to use several tools. One tool is used to load molecules from the protein data bank and generate surfaces from them. Another tool is used to position and orient the molecules with respect to each other. This is made particularly challenging by the 2D nature of the interaction, by the lack of collision detection to determine when the molecules are touching, and by the lack of 3D depth cues. The SketchBio project provides an integrated 3D environment for loading and posing macromolecular structures. Its natural 6 degree of freedom, two-handed manipulation, along with collision detection and shadow plane depth cues, let us finish in 1/5 the time. Responding to the needs of our collaborators, SketchBio also has custom behaviors for building complex molecular structures. A crystal by example mode supports the rapid construction of extended structures, such as clot fibers, microtubules, and the actin filaments shown here. First, the base structure is replicated the desired number of times. Next, the first two replicas can be grabbed and moved relative to one another to form the extended structure. Collision detection and live update make this process much easier than trial and error approaches in other tools. The entire collection can be copied and pasted, producing a set of actin fibers that can be independently animated. To bring objects into close contact without touching, springs can be used along with collision detection. These can either pull objects to a particular location in space, or, as shown here, pull two particular locations on different molecules into contact. Tooltips appear to help the user remember the order of multi-step operations. Multiple copies of the vinculin molecule are made to provide actors and extras in the video. Collision detection also works with direct two-handed manipulation to help the scientist place molecules into close contact. Here, vinculin molecules are being attached as bridges between the actin filaments. This is an example of fine manipulation of objects in close contact. Keyframe animation is supported to enable the generation of specified motions to help understand and explain concepts. Here, we're building the animation backwards, so that the fibers come together at the end of the animation. We position all of the fibers and vinculin away from each other at the beginning of the video. Because it takes so long to render a high-quality video, a live preview from the moving camera's point of view is available. This lets the scientist make sure that the resulting motion is correct before launching an offline rendering. Now that the video is ready, it can be sent to Blender with the press of a button. SketchBio is not just about making videos. The interactive 3D interface, combined with collision detection and springs, makes SketchBio a 3D thinking tool so scientists can explore proposed interactions to see which ones are feasible. It's being used by Susan Lord's lab to explore fibrin protofibrils and by Sharon Campbell's lab to explore the impact of vinculin on the structure of actin fibers.